This is the story of Chilika, a 1,000 square kilometer lake on the east coast of India. Because of its connection with the Bay of Bengal, Chilika is also a lagoon. And like all coastal lagoons, its waters are rich with life. In the late 1990s, Chilika was a weed-infested, dying lake. But thanks to ecological restoration by the Odisha government, it's once again a steady source of livelihood for thousands and a vibrant refuge for wildlife. For threatened wetlands everywhere, Chilika's resurrection is a story of hope. Chilika is one of Asia's most important wintering grounds for migratory birds, and greater flamingos are among the earliest visitors every winter. They are believed to come here mainly from the Kutch region of Western India, where they breed. For them, the journey to Chilika is perhaps not such a difficult one. Other migrants are known to travel thousands of kilometers to get here. Many come from as far away as Europe and Central Asia. Chilika's waters abound with food, and for these Eurasian widgeons, it's time to replenish their reserves after a long journey. Not all of Chilika's birds are migrants. Some are permanent residents, like the little cormorants. When these expert fishermen gather to hunt, others follow, hoping for a stray morsel. A gull will even pretend to be a cormorant for a piece of the action. Many species of birds can feed simultaneously in the lake, thanks to different diets. The shape and size of their beaks and legs allows them to exploit different resources. The secret of Chilika's biodiversity lies in its mix of salt water from the sea and fresh water from rivers and streams. This freshwater to saltwater gradient creates ideal conditions for life. Every high tide brings rich nutrients from the sea into the lake. At low tide, the exposed mudflats attract hungry plovers and sandpipers. For crabs, the mudflats are not just home, but also an important source of food. Hidden in the substrate are microscopic organisms, which the crabs extract with astonishing efficiency. The crabs must hurry, for in a few hours, the tide will be back. Seawater is Chilika's lifeblood. Without a regular inflow, the lake could die. In fact, from the 1970s to the 90s, Chilika went into a decline when its sea inlets became so choked with silt that seawater was reduced to a trickle. At the same time, inflows from streams and rivers began changing the lake's character, from brackish to fresh. Invasive freshwater weeds spread rapidly, soon covering a third of the lake. With the water spread shrinking every year, fishing activity and fish catch plummeted. Chilika became known as a dying lake. Scientists studying ways to restore the lake's health decided that urgent action was needed. So in September 2000, after rigorous modeling studies, a new mouth was dredged open between the lake and the Bay of Bengal. With seawater flowing in and out again, there was a positive transformation. As the lake's salinity levels increased, freshwater weeds retreated, and open water was restored. Fish populations recovered, and catches went up significantly. Today, Chilika is a reliable source of livelihood for over 200,000 people. The fisherfolk of Chilika have devised dozens of different methods to catch the lake's fish, prawns and crabs. Apart from the active fishing that people do, passive fish traps are at work 24 hours a day. The intensive fishing by humans makes life difficult for one of Chilika's top predators, the white-bellied sea eagle. With its 7-foot wingspan and keen eyesight, the eagle is a master hunter. It's a case of so near, yet so far. The eagle cannot risk getting tangled in the net. It must look elsewhere for a meal. One fish eater that has learned to take advantage of the nets lives underwater, the Irrawaddy dolphin. These beakless dolphins are more closely related to killer whales than to other dolphins. Chilika is home to about 150 of these highly endangered mammals. This species employs a unique spitting behavior while hunting. The intelligent animals have learned
Okay, sir. On behalf of the organizing committee, I welcome our first speaker, Professor Shapan Datta. So, sir, could you just try to share your, just to check before we go into the.
Hello.
Yes, yes, sir.
Should I just
Share screen. Yeah.
share screen, right?
Yeah.
Good evening, sir.
Good evening. And then share. I again put share.
Share your the PowerPoint. Yeah.
Yes.
Yeah. Oh no, not this one. You have
No,
to
no,
select.
not this one.
No.
No, not this one.
Yeah. So you just stop share from the top. Stop share from the top of your screen. Yes. And again, share screen.
So just a minute. Just open your PowerPoint and minimize it.
Is your PowerPoint open, sir?
Okay, I can do that.
Open your PowerPoint on the desktop, minimize it, and then share your screen, sir.
Okay. Yes. And minimized now. Yes.
Then share your screen.
And then share. And next.
Next, you have to select the PowerPoint that is visible there.
But I think you have not yet shared, sir.
Okay. Should I? Has it done now?
Yeah,
yes, it has done.
Okay.
So can we start, sir? Dr. Kutu, please.
I'm ready.
Sir, Patak sir, what should I do? Should we wait?
Dr. Patak sir, I think you will introduce.
A few minutes, 2-3 more minutes.
Yes, sir. Okay.
It's not yet 7.
No,
Don't
sir,
rush.
it is not yet 7.
Not
Yeah,
yet 7.
yeah, we should not start early.
Kutub is too excited and excited young scientist.
Or just I don't want to have a technical problem during the session. That's all.
No, I'm fine. My sound is all right?
Yes, sir.
Looks like Dr. Maiti is getting very expert on handling all this.
This has become so frequent. On daily
online-
basis, there are meetings.
Dr. Maiti exactly knows where I stand, what I'm doing, and he just suggested exactly.
That's what initially I faced.
Okay.
So I learned from doing mistakes and
Actually, sir, great men think alike and they do alike.
It's great.
I think, Kutub, now we can start.
Yes. Sir, so yeah, it's 7:02, so I'm starting formally. So a very good evening to everyone.
100.
Sir?
Yeah, go ahead, go ahead.
Okay. So a very good evening to everyone. You know what I should say in this evening session, that this COVID-19 is actually democratized the science. You know, we are reaching wider and diverse participants and who are seeking for scientific knowledge. So this is a great way to be connected with the audiences. So from the morning to great deliberation in this first Indian Rice Congress that is being organized by Association of Rice Research Workers and National Rice Research Institute, Cuttack. So we had inaugural session and then plenary lecture and then panel discussion in the first half of this day. And in the second half, we completed just 6 concurrent sessions. That was a very tough job, but we could complete it successfully. And now it's time for another very large job, because you know a lot of participants showed interest for this special distinguished evening lectures. We have 2 speakers, and we would be knowing about the speakers from the 2 co-chairs of this session. One is Dr. Dipankar Maiti. He is the director of National Rice Research Institute, Cuttack, and another co-chair is Dr. Himanshu Pathak. He is the director of Niyazam Baramati, and he is also the secretary of Association of Rice Research Workers. So it's to you, sir, both the co-chairs.
Is president of ARRW?
Sorry.
So thank you very much. Now it seems Kutub is really very busy for the whole day. Thank you, Kutub. Thank you very much. Good evening to all of you, and maybe good morning to some of our colleagues who are joining from outside. It is indeed a great pleasure to welcome all of you in this evening lecture. And as you know that the whole day, we really had a very exciting time, very good deliberations. And I was also happy to see that Professor Datta was also there in our inaugural session. And now it is time to listen from him. Professor Datta is known to all of us. He has been mentors to many of us who are there in the audience today. So therefore, he really doesn't need any kind of introduction. But for many others who are students or just joining, let me say a few words for introducing Professor Swapan Kumar Datta. Professor Swapan Kumar Datta is a well-known plant scientist with vast experience in plant biotechnology, agricultural science, and global agricultural policy. Currently, he is holding a very prestigious DBT Distinguished Biotechnology Research Professor at University of Calcutta. He has served in different distinguished positions. He was Vice Chancellor in Vishwabharati University, Santiniketan, West Bengal. Also Deputy Director General Crop Science in Indian Council of Agriculture Research, New Delhi. Before that, he was Rasbihari Ghosh Chair Professor in University of Calcutta. Then, of course, very distinguished other positions, including Senior Scientist and Harvest Plus, Global Rice Crop Leader at IRI, International Rice Research Institute, Philippines, Visiting Faculty at UC Davis, USA, and of course, FMI Fellow at Basel, Switzerland, Senior Scientist at Zurich, Switzerland, and DAD Fellow, and several others. Among his most known contributions, Professor Datta and his team developed genetically engineered rice, that is golden indica rice, high iron rice, bacterial blight and seed blight resistant rice, single microspore derived embryogenesis, and double haploid rice. And recently, very recently, his group developed efficient genetic transformation system in chickpea, pigeon pea, and jute. His group's work on GM hybrid BT rice is well established in China, and there are many other materials waiting to be released. He has been the recipient of several prestigious awards, several of them. He is fellow of all the national science academies of this country and also few others abroad. He has supervised 38 PhD students and published around 200 research papers, which appeared in very prestigious journals, including Nature Science, Nature Genetics, Nature Biotechnology, and many others. Professor Datta has been named as one among the top 25 Indian scientists from all fields of science by India Today in 2011. And very recently, he is one in 2% top global ranking scientist as per one of the study by Stanford University. With these few words, it is indeed a great privilege, great honor for me to invite Professor Datta to deliver this evening lecture. Sir, the screen is yours. Professor Datta, please.
Thank you, Professor Himanshu Pathak, a good colleague and friend of mine, and who himself is a distinguished scientist. I've been knowing him for many years. And today, in this evening, also I see Dr. Dipankar Maiti, and a very distinct and a very energetic scientist, and who have been my PhD student working at Calcutta as Kutub, and also fellow scientists all over India as well as abroad. Good evening to everyone who are attending today's evening lecture. I'm very much thankful to the organizer, Secretary Dr. Maiti, and President Dr. Himanshu Pathak for giving me the opportunity to speak to you, and also the Association of the Society Arrow and NBRI for arranging a wonderful meeting that I've been listening from morning today. The morning inaugural session was fantastic with speech by Matthew Morrell, Dr. T.R. Mahapatra, the DG ICAR, as well as the panel discussion. I've been listening to many distinguished scientists of India, Dr. T.R. Sharma, Dr. Arvind Kumar, Dr. A.K. Singh, and many others, a big list, and also other participants. I think after a long time, even during COVID situation, we have been at home, but today RISE meeting brought me again alive. RISE is live, we know that, but getting that fantastic RISE meeting, I've been thrilled and excited to be part of it. Thank you once again. Thank you very much for arranging a wonderful RISE meeting. My topic been given that I have to talk on nutrition-rich rice, and that's the title I have given. And hopefully, I will try to give the essential material and information on the subject. Very briefly, to bring that information in perspective, as we know that already, the 4+ billion people live on rice. It's such a crop, so much nice to work on that crop. 158 million hectares annually. My sound is okay?
Yes, it is
sir.
fine.
Yes,
Perfectly fine.
Okay, thank you. 158 million hectares annually, producing 700 million tons. 90% of this rice grown and consumed in Asia. India's scenario is 43 million hectares producing 117 million tons, 11 million tons exporting, and 4 to 5 million tons surplus goes for storing. This is India's status. Interesting part is 800 million people gets 5 kg rice per person per month, which is basically free. That means we have to produce enough rice to make that government policy and support to this. But Ramesh Chand today made a cautionary note that cost of rice production economics is not sustainable. Whether I put a question, whether it's a sustainable on economics aspect is important or social aspect is important. Whether we consider more social aspect and produce more rice and provide the people who need them. I would go on the second comment that what I made. The interesting part is, in spite of the total rice production and rice availability, only 4% goes to the global market. So any given year or 2 years, if there is a rice production for some reason get reduced, there will be a global crisis like COVID-19. So we have to be very careful when a crop, one food crop that feeds for 4 billion people, we cannot have a complacent that we have done enough and we don't need to do more. Rather, I would consider when we are doing good science in rice, large number of scientists actually from India are there, so actively participating in rice research. We must continue doing more rice research and to, in every aspect of rice, either our yield or other traits or biotic or abiotic stress, and of course, nevertheless, nutrition would play an important role. And from the market point of view, the $200 billion market, where 90% only consumed in Asia. So that means this is a crop so much of important, but it lacks nutrition. Meal rice lacks nutrition. So today's my talk would concentrate on how we can improve nutrition and what is the status and how we can address that issue. Again, I don't need to talk on this particular slide. Everybody know this, all the data that shown in this slide. But one point is important, 35 to 43% children and women in India are malnourished. No matter how much we are producing many other food crops, etc., but we still are suffering. Our food habit and particularly rice lacks nutrition, and that perhaps one of the attribution that we have lots of malnourished children and women are suffering. And from a calorie and from protein perspective, that If you consider rice and wheat together, it's more than 70% calorie comes from this cereal, but only rice from India is around 30-35% that comes from rice, and which is already a lot considering 1.3 billion population. But many other Asian countries, if you see this slide carefully, Cambodia, Myanmar, Laos, Bangladesh, Vietnam, Indonesia, lots of calorie that comes only from rice. So that's a very important point. But with the nutrition or malnourished children and women, the entire scenario, if you see this slide, it's very, very alarming that baby gets low birth weight, child stunted, adolescent stunted, pregnancy, low weight gain, women get malnourished, and all of this chain system, they're all linked to the lack of nutrition, as I mentioned, as malnourished children and women. Now, bioengineered plants. To me, the conventional breeding is much more bioengineered than the genetic engineering or genome editing. I think Dr. A.K. Singh and many rice breeders, if you consider the way they're working, or our great guru, Dr. Gurudev Khush, and I would come to a few slides on that, and you would notice that so much of the genetic engineering plant breeders are doing, rice breeders are doing it. And only thing, they don't care of so much combinations, recombination is taking place. They look for a very precise trait and ultimately select with a certain period of time. But the conventional breed, in addition to conventional breeding, marker-assisted selection, genome, genetic engineering, genome editing, and all of these things together that we're addressing as bioengineered plants. So to me, plant breeding itself is a bioengineering, and only with the precision or with the certain sort of combinations and the philosophy behind the scientist that play a particular name, that how he want to do that. Nevertheless, by increasing the quantity of different nutrients, that is, one is the vitamins, essential amino acids, minerals, and phytochemicals, that we can increase it to get it nutrition-rich rice, or we can do another way by enhancing their availability, stability in plants, and bioavailability for the consumers. That I would come to a second part in a few minutes. Now, this is a tree. Look at this tree, ICAR-64 development. Can we imagine how much combination, recombination is taking place? Now, A.K. Singh can make a one slide, can show that how he is Pusha 1121 or Pusha, all of his material, that's what he has been doing, and doing miracle in India and making India proud. Then I'm sure there's so much of genesis, so much of recombination is taking place. This is Dr. Khush is showing that how many different races and cultivars and kind of land races are being used to develop one ICAR-64. Now, if this one, again ICAR-64, when we're making a genetic engineering, and then even we get a particular trait like golden rice or any other trait, then certain people will consider, oh, there are so many combinations. So that means everything linked to the genetic engineering. And so that's the reason I put two slides. One is conventional breeding and one is genetic engineering. Same ICAR-64, because we develop ICAR-64 for many traits, genetically engineered, and it's showing the different recombination. So, but at the same time, if the phenotype or the trait or other traits remains same biologically and functionally, and we have to only look for a specific desirable trait, what we want to introduce, and a certain aspect of that biology of that trait can be studied, and not looking so much of recombination taking place. If there is no change in phenotype, I think all the materials are acceptable, safe, as good as conventional breeding. Now, there is a review, Niraja's group at Hyderabad group, RISE group that published, and that that paper shows that rice somehow showing the minimum protein with compared to other cereals and other millets. Same with iron and zinc, and that shows that where is the problem with this particular crop from the nutrition point of view. Now this picture, now if I could have compared it now COVID-19, the worldwide COVID-19 is there. Now malnourishment also is everywhere, particularly vitamin A deficiency or anemic or iron deficiency. So both the trade, both the important component all over the world, particularly Asia, Latin America, Africa, etc., etc. And so it is definitely a very alarming situation. Now, someone could say that, well, there are good vegetables available. You can consume vegetables. There are certain people might say that there are lots of capsules, vitamin capsule. Nowadays, during COVID situation, most people are consuming lots of capsules of vitamin C, vitamin E, vitamin D, etc., to develop immune better. But at the same time, but that's the way people to address that issue. But nevertheless, children or many people cannot afford to consume that vegetables or tablets. So they, if their food habit is based on rice, they need rice. They need rice and rice with nutrition. So that's one thing is very clear to me, that we need to buy fortified rice with the nutrition components. Now take the example of vitamin A. So definitely the most dietary vitamin A is derived from plant source in the form of provitamin A or beta carotene. And after consumption, the body converts it to vitamin A in our body. So in general, carotenoids or this pathway are present in all green tissues. So it's basically this pathway is already there in the plant. So we just engineered it into the seed. I'll come in a minute. Now there are different aspects that we can see that how we can address that issues. One is screening for provitamin rich rice varieties, mutation breeding, transgenic approach, or trans-carotenoids, beta-carotene rice, or increased bioavailability. These are the main objective that we put it in place. Now, screening several hundred of rice germplasm available at ED, we screened for carotenoids accumulation or carotenoids presence. One of the material from Amarillo from Cuba that shows a lot of carotenoids, and even polished seed also showing a little bit of carotenoids as compared to some other material. But this screening is quite clear that beta carotene or that particular component is not available or cannot be seen after polishing rice. Now, there are several of the different types of rice available in India and elsewhere, and some people think that they might contain a lot of carotenoids, but it is not. They look different colored, but after polishing, we look for the carotenoids levels, and we did not find presence of carotenoids after polishing. Now, this is the sort of scenario that the green part IPP to GGPP, geranyl pyrophosphate, and this is the common pathway in plants. It presents all green tissue of the plant. Now, phytogen synthase, phytogen desaturase, lycopene cyclase, these three enzymes or three genes, when incorporated in rice endosperm with endosperm-specific promoter, then we ultimately find alpha and beta carotene, and that is the pathway introduced in rice to get the beta carotene rich rice or golden rice. Now, before coming to that point, here is one slide just in a recent one of the book chapter we have written, and where it shows the rough rice, brown rice, and polished rice. If you look, the iron 8.8 ppm to 4.1 ppm, so everything got reduced. And percentage-wise, and including vitamin B9, that is folic acid or folate, also got drastically reduced in the polished rice. So this is the scenario that what happens. But traditionally, this picture I have taken from Bangladesh. And earlier, during our childhood time, this was a common thing, teki chata, chal. We used to consume this very traditional system, where it contained a bit of semi-polished rice and contained a lot of nutrition, but today it is just ceremonial. It's not available anymore. So that's no longer can be feasible. Now, the concept of the golden rice or the carotenoids pathway that came at ETH Zurich, you can see Ingo Patricus, Peter Bayer, Karabi Datta, Mayank. So there are lots of people got involved in this particular work. And after perception or concept that developed at ETH Zurich, many laboratories across the world started that work in a commercial rice or a cultivated rice. And ultimately, we developed the golden rice in different indica background and several other Asian countries' rice background. And this is again another detailed such studies. We recently also published in a book chapter, but 2003, we showed the bioengineered indica rice with the carotenoids pathway that is can be shown and can be evident in many of the Asian rice varieties. Now here is this golden rice, and also see the 3:1 kind of Mendelian segregation very clearly shows that how these genes, a dominant gene, can integrate in rice and can function. Now this particular slide, I'm showing it, I would link to some of the expression level. There you can see that many of the independent lines that shows a single copy to multiple copies, and also truncated as well as tandem arrangement. And so far, we did not see that a particular, in general, the high copy number shows better expression, but in many cases, we also did not find that. So it's not whole true that it can be just directly correlated with that kind of expression with the copy number. Now, of course, what we did also, we used different marker gene, hygromycin, as well as PMI, phosphomannose isomerase. And interestingly, we found that we used two plasmid. One is selectable marker, and other plasmid containing gene of interest. And here you see that this is the CRID gene, and hygromycin gene is absent. Here also there are several absent. As we put plasmid, two plasmid separately, and we introduced in rice, so the next generation, we can segregate out the selectable marker gene and keeping the gene of interest, meaning that very simple way, by simple plant breeding, we can remove the marker gene from the gene of interest. And Vilas, while doing PhD, he did this work, he published this work. Now here, what I'm showing that there are different independent line, and they have a different kind of expression. And this expression, we could not correlate with the copy number. In some cases it works, in some cases it is not. So there cannot be any conclusion can be made on that, but nevertheless, we would definitely select the best expression level, and also if that is a simple copy integration, and that the event would take it forward. Now, Niranjan Vaishak in the beginning did one wonderful work that to see how anthraculture could make stabilizing the genome or stabilizing the trait. And with anthraculture, we showed that double haploid shows the stable integration and no segregation takes place. This is the perception that we wanted to prove, and we showed that it works. Now, later part, when we developed more indica rice lines, BR29, one very commercially available Bangladesh rice and other rice, indica rice from India. And one of them is BR29 that we developed again a homozygous material with anthraculture, and clearly shows the double haploid material setting seeds, and haploid does not show any seed development. And here is the carotenoids rich homozygous line coming from DH line. Idea was that because carotenoids accumulation degrade in course of storage, but if we have DH development and that it becomes more stable, phenotype remain unchanged, remain very intact, and for breeding purpose and for breeding aspect, this would be very useful. So one of the things Mayank, well, in his PhD work, what he was looking to see that storage and cooking, a different way of cooking, whether that can affect the carotenoid expression. And exactly, we found that yes, after cooking, we loses the carotenoids, total carotenoids, but many other 10 to 15% definitely is lost during cooking, but still it retained quite a substantial amount of carotenoids. One of the other aspects, Deepak Gayan, well, in his work, what he shows that downregulation of lipoxygenase gene that reduce degradation of carotenoids of golden rice during storage, because rice normally kept stored in an ordinary condition in a sort of jute sack and with the high temperature, and it has been found that very high level of carotenoids rich rice, they degrade carotenoids very rapidly, and within 6 months to a year, they reduced dramatically. So we, two ways we want to reduce that degradation. One is to develop homozygous material, and secondly, we want to down-regulation of lipoxygenase gene, because lipoxygenase enzyme actually make that sort of degradation. So that's the reason we want to down-regulate the lipoxygenase gene that reduce degradation, and it successfully showed that it works very well. Now I come to the essential minerals, particularly iron. The iron deficiency is the most widespread micronutrient deficiency worldwide. 30% world population really suffers from serious nutritional problem caused by insufficient intake of iron. Now, one of the important aspects that the iron, that it constituent of hemoglobin, the oxygen carrying component of blood, and also a part of myoglobin that helps muscle cells to store oxygen. And today, with the COVID-19 situation, that's the reason the hemoglobin levels is very important that we have to maintain. We have to keep a high hemoglobin content because they're the oxygen carrier. So that means today's iron-rich rice even could be very helpful to our human consumption, because if we have more ferritin, more iron, and that would help oxygen as an oxygen carrier. That would help definitely not only anemic situation, but also in the COVID situation. Now, some of the brown rice that shows high iron before polishing, but vacuum packed and a very different kind of processing system need to be maintained, as Dr. Mahapatra today was mentioning, that processing and this aspect of packaging we're not doing enough, and that's true. These are the material. Many countries they're available with high iron and other nutrition, but that it needs a different kind of processing system. But we lack that one in India. Now, as I mentioned already, the polished rice contain very little iron, so we can do two ways: by increasing the concentration of the iron binding protein, that is ferritin, or by reducing the amount of iron absorption inhibitor, phytic acid, that is low bioavailability of minerals bound to phytic acid, can cause iron deficiencies in human population, where staples like rice and wheat and maize are the major or only source of nutrition. So that means by increasing or by reducing the anti-nutrients. So here is the slide. Now, if you look this slide very carefully, you see that very quickly the different milling times that iron content get degraded or reduced within 30 seconds or 1 minute or 2 minutes. Almost iron is almost gone, and that's the reason we don't get any iron in our rice, or if we get it, very little iron. Similarly, like vitamin A rice, I mentioned, again, screening for iron-rich rice varieties, mutational breeding, and transgenic plant strategy that being looked upon. At the same time, to reduce the content of phytate in rice grain that could make more iron and more bioavailability of iron and vitamin A. Now, screening at ERI, at least several thousand material have been screened, but we did not. We found a lot of rice has high iron, but after polishing, nothing is significant. So there is nothing to talk about it. So that means this aspect is not helping to look for natural resources for iron. So iron incorporation, iron improvement has to be done by genetic engineering. Now the ferritin is the iron storage protein that stay inside the material, and that's the material that contains iron, and we need to incorporate ferritin or more ferritin in rice material. What exactly we did? The ferritin gene driven by globulin promoter or prolamine and several other endosperm specific promoter. Vasconcelos, one of my PhD student at ERI, what she did, she engineered rice with this combination of different genes with promoters, and showed 2 to 3-fold more iron after polishing the rice. So that shows significant improvement. Later part, much more work has been done at Calcutta University, and but we so far did not or could not make more iron content, although we have been looking at different approaches, including transporter gene, etc. But at best, we could make 2 to 2.5-fold higher iron content. But nevertheless, that itself is good, because after polishing, that iron stay there, and that is available for our consumption. Now, phytic acid, the anti-nutrient, and this is the things that we would like to remove from the rice. And already the low phytate canola is being commercially used in Canada and elsewhere, and that gives the cattle with more iron content, and similar things probably would be very useful to do it in rice. Now, phytic acid, as I mentioned, that strongly binds to mineral cations like iron, zinc, calcium, magnesium to form a mixed salt called phytate. That means this phytic acid binding with all these minerals, it causes non-availability of the material. But if you get a down regulation of these two enzymes, particularly this enzyme MIPS, and the second one is IPK1. Now, these two enzymes in the pathway of the phytic acid pathway, if we remove or if we reduce these two enzymes, resulting that these minerals would be freely available, and including phosphorus and iron, and that will be more bioavailable to our system. And what exactly we did by RNAi-mediated downregulation of myoinositol 3-phosphate synthase to generate low phytate rice. Second one is IPK1 gene that we used to make the rice with low phytate. And in recent time also, we have been looking several other genes, and publications are in press or coming or have already come. So we are still looking the same system and approaching that to get more bioavailable of minerals, including iron. Now, folic acid is another very important component or folates, which are benign vitamins and needed in almost all organisms because it's a very important one, but it related to the biosynthesis, nucleotide biosynthesis, amino acid metabolism, etc. And humans and other animals cannot synthesize folate, and plants usually being the main dietary sources. So something has to be improved. Plants with more folate, and there are certain works going on elsewhere, and even the genome editing can be approached to do more work on this particular aspect. Alamgir, a student from Bangladesh who has been involved, what he was looking that we have developed high iron rice and we have developed high beta carotene rice. Why not to combine both and to get a material that with high iron as well as high carotenoids? We did not move further, but this was achieved without any phenotypic or pleiotropic effect. After getting these two materials done by bioengineering, then simply plant breeding was done and developed the material. Now, one of the things that is very important, that synergism of the vitamin A, iron, and zinc. There are certain literature that clearly shows that if more vitamin A or more iron or more zinc is present, so one of the component, one is helping to absorb the other one. So that means more vitamin A can absorb more iron or zinc iron, and vitamin A, zinc, iron. So nevertheless, accumulation or enhanced level of vitamin A, iron, and zinc help in synergistic positive effect in the plant system as well as for our human consumption. One of the issues that lysine biosynthesis, normally what happened that from aspartate the dihydrodepicolinate synthase gene that help to stop the feedback inhibition, because more lysine in a general part, the more lysine is produced, they have a feedback inhibition and then they stop producing it. But once that this particular gene from Corynebacterium, if used, then it reduces that or blocks the feedback inhibition system, so the more lysine can be produced and then ultimately help. Preliminary work we have done, and we haven't published it because we didn't get many transgenic events. We want to improve it more, but this particular approach and concept has been published in several crops, including rice from China and elsewhere. So that is one possibility. It can be incorporated for a better improvement of better quality protein. But NRI, congratulations to NRI that they have done wonderful work, CR-THAN 310, that this group has made 10% protein, and now this particular approach they have extended to other popular variety, and so that means we can have rice with 10% protein, which is a remarkable work, I would say. And Dr. Mohapatra today was also mentioning there are certain rice varieties from Assam that has even 17% protein. So we should explore and look into those genetic resources, particularly for protein. And if we have this protein incorporated into high-yielding varieties, and that would be wonderful that to have an enhanced protein content in rice. Genome editing definitely today is a new technology based on transgenic research, and it's a revolutionary technology used to generate novel variations of existing genes in a species, but it has to work when or there is a efficient transformation system available, and then it can be incorporated in a better way. So both efficient genetic transformation system and genome editing could make a bigger impact in our future RISE improvement. Now, I think our next speaker will be a very good friend of ours, RISE scientist, Professor Pamela Rowland. So Pam Rowland has made one particular interesting work, that is removing the marker from the system using the particularly genome editing. And that way, this approach could be used to integrate any cassette of interest to a safe harbor loci. But the homology of directed repair is not that efficient, but that sort of things can be improved in future. But anyway, this is a good technology that has been reported and can be used very simply to remove the marker. And I think today morning, Dr. Mapat was mentioning about starch, branching starches and slow release of starch, particularly as we have been suffering from malnutrition. And in that way, any changes of amylopectin and incorporating WEXI gene and reduction of those things could enhance or could make a better better sort of composition of starch composition, and that not only for it would help our system of diabetic sort of issue. At the same time, it has a tremendous impact in the industry. Now, the iron, I just mentioned while I was talking about folate that iron-rich and folate-rich rice. So there are genes that OSVIT2, a vascular transporter gene, that it could increase the grain iron. So genome editing has a tremendous potential to address that issue and to develop that sort of rice in future. Now, protoplast, 1990, we first published the genetic engineering of indica rice. So 30 years gone. Protoplast still I love, and it's a fantastic system. And protoplast rice or any other crop now again is coming back. It's a great system for many transient assays like genome editing, efficiency, protein expression, subcellular localization, biomolecular fluorescence, complementation, etc. But regeneration, to me or to our team, was not difficult. It can be done. It takes time, or to maintain that embryogenic cell suspension is a key. But if it is coming from anthraculture-derived embryogenic cell suspension, then getting protoplast and regeneration to plant is simple, very simple. So if anyone want to use protoplast system, I think it's an efficient system, and it's very efficient in a way that any sort of vector, any cassette or any protein expression, or at the same time a large number of gene or variations can be generated and can be seen using a protoplast system. Because when you isolate protoplast, 10-20 million protoplast is nothing, and then for your tangent or stable transformation system, you don't need more than 1 million protoplast. So in one go, you can have 20 experiments. So this is such a efficient, and there are people started working on protoplast, and I'm glad that I feel very exciting seeing those kind of research that coming with genome editing using protoplast system. Now, one or two issues on GMO, particularly golden rice. When Ingo Patriciu's group published in Science paper long back, and after that, we published at least 10 research papers on different aspects of golden rice. India also, IRI, Dr. A.K. Sings and other people also made a lot of work. But even Ramesh Chand was mentioning today that golden rice is very important, but is not available to the commercially or in the market. Why? Question is why it is not available, and why we're not or deprived of having such a wonderful work and not available to the people. And what is the point of doing science? What is the point of be excited to be involved in science? And after having done the work, same with the high iron rice, low phytate rice, and golden rice, nothing is moving. And I just saw a paper which is not yet published from Bangladesh. They're in the final stage of field evaluation, and soon they would be field evaluated. And one person, it's not science, one person, Motihar Chaudhary, Agriculture Minister of Bangladesh, made such a big change, big differences. And hopefully someday our system will allow us to move on. But it is a very strange situation that we are unable to move with the product even developed for even a material like golden rice. Now this is my concluding slide. I come to my last part, 1 or 2 minutes I will talk. So the technology is neutral. Availability and use is subject to, I mentioned a few things, and you can imagine what I mean by saying that. So nice to see Pamela Roland. Hi, glad to see you after a long time. So a nutrition-rich rice, golden rice, high iron rice, low phytate rice, high protein rice, all I mentioned today is available, is done. The quality nutrition-rich rice will have health benefit to all, irrespective of income group. Today, in COVID-19 situation, we need all high nutritious food, and rice can provide those kind of things is wonderful. It would boost our immunity. Market demand of high quality rice will have benefit to the rice farmers, industry, and economic gains for all. And that's the reason my title is to all. It's not just malnourished people, but everyone, the farmers and everyone. Too much regulatory restrictions plus political and government will, and the policy needs serious attention to make the products available to the people who need them most. I think we're struggling with this policy on the policy matters, and this needs serious attention. So final conclusion, nutrition-rich rice for all, it's a reality. As I mentioned, protein, vitamin A, iron, low phytate, delivery possible with collective will and government policy. That's thank you. This is some of the people who are still working a few years back. And when Malcolm Elliott was visiting our lab at that time, it's I believe 7 or 8 years back, and that was this big chat. And I see Kutub also standing here. Thank you very much. Thank you for giving me this opportunity to talk to you. Thank you.
Thank you, sir. Thank you very much. It was indeed a very exciting talk. Of course, you raised a few very critical issues. I think at some point of time, somebody has to answer those questions, and hopefully those questions will be answered. But sir, what we plan that we shall take questions from our audiences maybe at the end of both the presentations.
Right.
And my friends, if you have got any question or query, you can put it in the chat box so that it will be easier for us to take them all together. And we will be requesting both of our speakers to respond to those queries. So thank you very much.
Okay.
Friends, it is indeed a great honor to introduce the next speaker of the evening. I think for morning for madam, but for us it is evening. It is around 8 o'clock going to be. Friends, Professor Pamela Ronald is recognized for research in infectious disease biology and environmental stress tolerance. For isolation of rice XA21 immune receptor in 1995 and of a novel microbial immunogen in 2015 revealed a new mechanism with which plants and elements detect and respond to infection. She is also known for her leading role in isolation of the rice submergence tolerance 1 gene. Her research facilitated the development of yielding sub-1 rice varieties grown by now more than 6 million farmers in India, Bangladesh, and even few other countries. Professor Ronald graduated from Reed College with a BA in biology, from Stanford University with a MS in biology, from Uppsala University with a MS in physiological botany, and from UC Berkeley with a PhD in molecular and physiological plant biology. She was a postdoctoral fellow in plant breeding at Cornell University and joined the faculty of UC Davis in 1992. In 2019, she was awarded an honorary doctorate from the Swedish Agricultural University. She is a faculty scientist in Berkeley Lab's Environmental Genomics and Systems Biology Division and a key scientist at the Joint Bioenergy Institute. She is a faculty affiliate of the Center of Food Security and the Environment at Stanford University. She was named a National Geographic Innovator and one of his 50 innovators who will lead us towards a more sustainable future. She was named one of the world's most influential scientific minds by Thomson Reuters and one of the world's 100 most influential people in biotechnology by Scientific American. She is a member of the National Academy of Sciences, USA. She is also a co-author of a very famous book, Tomorrow's Table, Organic Farming, Genetic, and the Future of Food. Tomorrow's Table was selected as one of the 25 most powerful and influential books with the power of inspiring college readers to change the world. Her 2015 TED Talk has been viewed by 1.7 million people and translated into 26 languages. She founded the UC Davis Institute for Food and Agriculture Literacy to provide the next generation of scientists with the training they need to become effective communicators. In 2019, she was awarded the American Society of Plant Biologists Leadership in Science Public Service Award. Madam, it is an honor to welcome you and listen to you in this evening. Thank you very much for accepting our request. Thank you for your acceptance and of course, going to enrich us with all of the exciting works which you have done. Thank you very much, Madam, and now the screen is yours. Over to Professor Pamela Ronald.
Well, thank you very much for inviting me to this meeting. I'm really glad I have a chance to participate. And of course, that nobody likes this pandemic, but at least we can see each other over Zoom. And I am going to ask you now if you can see my slides.
Yes.
Yes.
Please put it in the present. Yes. Great.
Okay. And thank you, Kudubudin, also for inviting me. It's great to see some old friends and to be able to participate today. So today I'll describe right genetic and plant purity. But first, I'd like to put this topic in the context of global demand for food and the current and future constraints on food production. Well, as you know, the human population is projected to reach more than 9.7 billion people by 2050. So in addition to having more people on the planet, we are consuming more calories than we have done in the past. This means that we need more food. Some estimates indicate that if we are unable to reduce food waste or alter our consumption patterns, we will need to double the yield of the major crops in the next few decades to keep up with food demands. At the same time that we have more people to feed, we are losing arable land. The United Nations estimates that the world loses 23 hectares of arable land every minute. And that's often through drought, desertification, and urban sprawl. That's what we see a lot of in California, and uncontrolled grazing. Now, as the population grows, there's already growing competition for fresh water, and fresh water availability per person has decreased fourfold over the last 60 years. This means that the projected doubling of food production must largely take place on the same land area using less water. Compounding these challenges is that many areas of the developing world, 60% of potential crop yield is lost to disease and environmental stress. Climate change presents new challenges, including rising temperatures and a higher incidence of pest diseases, droughts, and floods. Now, climate and agriculture are intertwined. Climate change increases the pressure on food production, while agriculture and land clearing for agriculture contributes to greenhouse gas emissions. Because of these challenges, food security remains out of reach for people in many regions of the world. According to the Food and Agriculture Organization, almost 690 million people went hungry in 2019, up by nearly 60 million in 5 years. So how can we address food security? And that's what I want to talk about today. Now, if the food system is to withstand climate change and sustain a growing population, it not only needs to reduce its own emissions, but it must become more resilient and efficient. Crop genetics improvement will be a key component of enhancing resilience in the food system. Today, I'll describe 3 projects that my lab team and collaborators have worked on over the years with the goal of enhancing resilience in rice, a staple food for more than half the world's people. This includes disease resistance, flood tolerance, and nutritional quality. First, I'll describe a project aimed at understanding the basis for disease resistance in plants. Now, this is an image of a rice leaf infected by the bacterial pathogen Xanthomonas oryzae-pathovar oryzae, the causal agent of bacterial blight disease. You can see Xanthomonas oozing out of the leaf. It produces this distinctive yellow pigment. This is the most serious bacterial disease of rice in Asia and Africa and can reduce yields up to 50%. To minimize losses, farmers rely on rice varieties that carry genes for resistance. This approach has been used for over 100 years. Well, when I started my research on rice, no one knew what these genes actually were. And it wasn't until the 1990s that researchers around the world finally uncovered the genetic basis of this resistance. Now, the resistance trait that I'll tell you about was discovered about 50 years ago in a wild species of rice in Mali. Plant pathologists showed that it was resistant to all strains of the bacteria tested. In the 1990s, International Rice Research Institute plant breeder and World Food Prize winner Gurdjieff Kush and colleagues mapped their trait and named it XA21. His team showed that XA21 confers resistance to all strains of the bacterium that were known at that time. Now, I was very fortunate to meet Gurdjieff at the International Rice Research Institute, and he shared a bag of seed with me. And my lab team then used map-based cloning to isolate the gene conferring the resistance trait. We engineered the candidate genes into a rice plant that is normally susceptible to bacterial infection. You can see that the leaves from plants engineered with the XA21 resistance trait shown on the bottom are remarkably more resistant to infection than the leaves from the conventional variety shown on the top. So to inoculate the plants, we take scissors, we dip in a bacterial suspension, and we clip the top of the leaves, and we wait for 2 weeks for the infection to score the infection. We then sequenced the gene and found that XA21 encodes a receptor kinase with an extracellular leucine-rich repeat domain, a transmembrane domain, and an intracellular kinase domain. Now, the predicted structure of XA21 suggested that the leucine-rich repeat domain would recognize the microbe or some molecule produced by the microbe and then transduce the response via the kinase domain. So what was a really exciting time, because in 1996, Jules Hoffman's group showed that the toll receptor in flies also was a key receptor for the immune response. And then in 1998, TLR4, the toll-like receptor 4, was shown in mice to also be a key component of the innate immune response. So it turns out that XA21 is broadly representative of this large class of plant and animal receptors that recognize and respond to conserved microbial molecules. They all have extracellular leucine repeat domain, a transmembrane domain, and in the case of the animal receptors, they associate with an intercellular kinase domain of the same class as XA21. So not only does this plant work have important applications, but it can also inform a basic understanding of how diverse organisms recognize and respond to infection. So we next wanted to identify the microbial molecule that activates infection. And we found that the bacterial pathogen Xanthomonas secretes a small sulfated peptide hormone named RAX-X. And that's shown here in the bottom of the bacterial cell. RAX stands for genes in the microbe that are required for activation of XA21-mediated immunity. And we had found a small cluster of genes that were required for activation of XA21-mediated immunity. And one of them had a long leader sequence and had a predicted cleavage site and a predicted sulfation site. We have been able to show that this protein, RAX-X, is cleaved at that site, and then it's cleaved by what's called the C39 protease domain of an ATP transporter. And that mature RAX-X is secreted outside the cell. So it's a sulfated peptide of 16 to 21 amino acids. So we were able to show through this genetics and proteomics works that XOO secretes this small molecule, and also that RAX binds the XA21 receptor, which is shown here in red. We also isolated a co-receptor called OSCIRC2, shown in blue. And this co-receptor is required for XA21-mediated resistance. Binding of this receptor ligand co-receptor complex leads to production of reactive oxygen species, ethylene production, defense gene induction, and ultimately a very robust resistance response. Now, this timeline summarizes 25 years of resistance gene cloning, and the colors represent different mechanisms. Now, the cloning of the first gene was in 1992, that was HM1, but you can see there's just been this incredible increase over the years. And XA21 falls into a class of resistance genes that encodes a receptor that directly binds a ligand, and that's shown in dark blue. And there's relatively few of these receptors that have been characterized to date. In this study, many of the other receptors shown here also have not been characterized. I mean, we estimate in rice that there's at least 100 receptors of the XA21 class, and only so far, most of them have not been characterized. So there's a huge amount of research for rice researchers to explore the function of these receptors and identify the ligands. Now, understanding the molecular mechanisms is vital, as it can facilitate transfer of these traits to other species, as well as develop strategies for rationally engineering these disease-resistant genes, and perhaps extending their recognition spectra outside of what is found in nature, and engineer plants for resistance to diseases which currently we have no method to control. Now, these studies have shown that immune receptors in plants and animals are remarkably similar. And we have also shown that the bacterial pathogen secretes a sulfated peptide that binds the XA21 receptor. Now, efforts are underway in my lab to engineer plant receptors to recognize a broader spectrum of microbes. And also other labs are working on this as well. Okay, so as I mentioned, climate change adds new challenges for farmers. So these kids walking through their family's rice paddy after a flood are not going to be able to harvest rice in the center of this field. That's because although rice grows well in standing water, most rice varieties will die if they're submerged for more than 3 days. The International Panel on Climate Change predicts that floods will increase as the climate changes. 25% of the world's rice is grown in flood-prone areas. In South and Southeast Asia, 4 million tons of rice, enough to feed 30 million people, is lost every year to flooding. This region is home to 70 million rice farmers, many who live on less than $3 a day. Now, rice breeders have long known of an ancient variety of rice that can tolerate submergence. So it was found in Orissa. And of course, the International Rice Research Institute has a tremendous collection of very valuable rice varieties. In the 1980s, breeders used a conventional breeding approach to introduce this flood-tolerant trait into rice varieties. However, the resulting rice varieties were rejected by farmers because the breeding dragged in undesirable traits such as lower yield. Now, at that time, breeders did not have a way to introduce the trait more precisely. In 1995, we began a collaboration with David McKill and Kinong Xu, shown here, to introduce the trait more precisely. So Dave and Kinong in 1996 had mapped this trait to chromosome 9. And in 1995, we began a collaboration to isolate the gene that we called submergence 1 or sub 1. We were able, after quite a long time actually, able to isolate the gene encoding the submergence trait. And again, we used this bat-based cloning approach that we had used successfully for isolating XA21. So on the left is the conventional rice variety that was grown for 2 weeks, submerged for 2 weeks, and then allowed to recover for 2 weeks. On the right is a rice plant that we genetically engineered in my lab with the new gene we named sub1A. You can see that the sub1A genetically engineered rice plant survived the flood in my lab really quite well. And we were quite amazed and excited that a single gene could have this robust effect. So at this point, we were certain that we had identified the gene that made the plant tolerant of flooding. But what was the mechanism? And of course, would it work in the field? So I've been lucky to know and collaborate with a really talented group of researchers that they began to uncover the mechanism which with sub1 enables the plants to endure complete submergence for prolonged periods. Now, it's been known for quite a long time that submergence creates a low oxygen environment and accumulation of ethylene. And in the absence of sub1A, a plant elongation signaling pathway mediated by the plant hormone gibberellic acid causes plants to grow rapidly, which depletes their carbohydrate reserves, and the plants die. Now, we had noticed that sub1A encoded a predicted ethylene response transcription factor. And in fact, ethylene, Giulio Bailey-Serra's lab and Takeshi Fukao showed that ethylene upregulated sub1A. And then it was shown by Ming-Che Chi's lab that sub1A, that a low oxygen environment can stabilize some other ethylene response transcription factors that we had identified named ERF66 and 67. And he was able to show that these transcription factors are stabilized during a low oxygen environment. And in fact, they're direct transcriptional targets of sub1A and are upregulated by sub-1A. ERF66 and 67 directly or indirectly activate expression of submergence responsive genes. And then sub-1A also downregulates carbohydrate metabolism and induces ethanolic fermentation. Sub-1A also downregulates GA signaling, and the resulting reduction in cell elongation and carbohydrate breakdown conserves the shoot meristem and energy reserves until the flood subsides. And together, these traits contribute to submergence tolerance. So does this trait work in the field? Of course, this is the most important question, and the answer is yes. Now, please watch this 4-month time-lapse video. It shows a sub-1 variety developed at the International Rice Research Institute using marker-assisted breeding. On the left is the rice variety with the sub-1 gene, and on the right is the conventional variety. At the beginning, both grow very well, but then the field is flooded for 17 days. And you can see that the sub-1 varieties recover rapidly from the flood. And in fact, the sub-1 variety produces 3-fold more grain than the conventional variety in these experiments. Now, I love this video because it shows the power of plant genetics. In 2008, our team was fortunate to visit farmers' fields in India and Bangladesh who had planted the new variety called Swarna sub-1. This image shows the fields after a natural occurring flood. You can see that the Swarna sub-1 variety grew very well. And in fact, it had a 60% yield advantage compared to the conventional variety called Swarna. Now, another group of, and I should say on this trip, we were fortunate to visit colleagues at Katak, and really were very impressed with the breeding program and all the work the group did there to carry out these field trials. In another group of researchers at UC Berkeley carried out a field experiment in Orissa, so in eastern India, shown here, and they wanted to see who actually benefits from this technology. In red are shown higher elevation areas, and in yellow are lower elevation areas, heavily occupied by people belonging to lower caste groups. Now, each of those black dots is a village that the researchers, including Kyle Emmerich, who led this work, met with the farmers there. And the researchers were randomly divided into 2 groups. So a treatment group where farmers were provided with 5 kilograms of Swarna sub-1 seed, and a control group where farmers were offered Swarna sub-1 seed, or they decided to grow their chosen varieties. Most often they grew just Swarna. Now, their investigations confirmed early results of IRI, that there was a huge yield advantage in the Swarna sub-1 variety. So every year there's been flooding, and they saw a 45% yield advantage. And as I mentioned, because the flood-prone region is occupied by people belonging to lower class groups, they are obviously seeing the marriage. So this study showed that sub-1A disproportionately benefits the world's poorest farmers, and many of these farmers who have generations of families grown their crops on flood-prone lands. Now, my third example is nutritional quality, and I know Professor Dada talked to you a bit about golden rice, and a lot of this work was begun by Professors Inga Petraikas and Peter Beyer. And you know that this project engineered rice to produce beta-carotene in the seeds, which is the precursor of vitamin A. And really the goal of this project is to reduce vitamin A deficiency among children and young mothers. It's estimated that 500,000 children go blind every year from lack of vitamin A, and half of the children will die. Rice varieties carrying golden rice accumulate beta-carotene in the grain, and so it gives rice this golden color you see here. And scientists predict that consumption of golden rice will save the lives of thousands of children living in regions where rice is the major food source and vitamin A deficiency is prevalent. The golden rice has been approved from consumption in several countries, but has not yet been released to growers for production. And as you heard, the regulatory process has been slow. Now, one limitation to conventional genetic engineering is that the location of the insertion of the gene in the genome is random. And the randomness of the insertion means that sometimes the transgene cassette will insert in a gene that's required for growth and development as shown here. So in this case, the integration of the golden rice transgene disrupted a gene required for plant growth, leading to dwarfism and reduced yield. And this also was another slowdown in the project, the golden rice project. But of course, breeders and scientists have overcome this, and they have another line that produces very well. So I'm just showing this to remind you that in general, when we use genetic engineering, we can't predict the location. Now, one approach to overcome limitations of conventional genetic engineering is to target insertion using CRISPR-Cas9. Now, this strategy was developed by teams led by researchers shown here, including Emmanuel Charpentier and Jennifer Doudna, who just last month were the first. Well, in 2012, they were the first to propose that CRISPR-Cas9 could be used for programmable editing of genomes, and who were awarded the Nobel Prize just last month. Now, CRISPR genome editing takes advantage of Cas9's ability to induce targeted DNA double-stranded breaks at specific regions in the genome. And the subsequent targeted insertion of donor DNA at these chromosomal double-stranded breaks can occur using an endogenous repair pathway. So you end up with the DNA inserted at a precise location in the genome. Now, Oliver Dong in my lab, who's now professor at Nanjing University, decided to use the CRISPR technology to insert the golden rice cassette into target sites in the rice genome. So for these experiments, we first needed to identify genomic safe harbors in the rice genome. Now, genomic safe harbors are sites in the chromosome that can accommodate transgenes without adverse effects on the host organism due to genome disruption. So it's also important to make sure that the inserts don't interfere with the endogenous genes and can be expressed properly. Oliver was able to target insertion of a 5.2 kb DNA fragment encoding key genes for beta-carotene biosynthesis, and he synthesized this cassette based on the pioneering work of Ingo Petrikis and Peter Breyer. And he was able to insert this insert at 2 independent golden safe harbors in rice. These rice plants produced grains that had high yields of beta-carotene in the rice seeds, and they also had normal morphology and yield. One of the useful applications is you can more easily stack genes. So in the past, when we're trying to stack genes that are in different locations in the chromosome, it takes quite a bit of efforts of breeding and selection. Genes are stacked in the same place. We can then use these lines for breeding in quite a simple way. And one application has already been shown in the sense that the disease resistance proteins, which are shown here, nucleotide leucine-rich proteins, are very important for the defense response, and they've been crystallized. You can see that nice structure of the ZAR1 resistosomes here. And I just wanted to highlight work of 2 groups that stacked 2 genes for rice blasts into the rice chromosome after actually quite a bit of work, because these were not inserted at the same location, and they were able to get a broader spectrum resistance than either gene alone. And similarly in potato, a group was able to stack 3 genes for resistance to Phytophthora infestans, which is a very serious disease of potato. And we're able to obtain potatoes that were resistant to several different strains of late blight. So we already know that stacking genes is very useful. And if we can target these genes into the same site in the chromosome, we'll be able to carry out this breeding easier. Now, I hope these examples give you a sense that it's really an exciting time for plant biology and agriculture. And I'm confident that the global agricultural community will continue to make important advances in plant genetics and breeding that really will enhance the resilience of our agricultural system. Yet, of course, we still have plenty of work to do. We need not only to work hard to advance the basic science, but we must engage the public in the work that we do as well. Society is only going to embrace the scientific advances if they are well informed and trust that the scientific process is transparent and reliable. So as agricultural scientists, we know that over 25 years of careful study and rigorous peer review by thousands of independent scientists, every major scientific organization in the world has concluded that the genetically improved crops currently on the market are safe to eat, and that modern methods of plant breeding are just as safe as older methods of genetic modification. Now, these are precisely the same organizations that most of us trust when it comes to other important scientific issues, such as global climate change and the safety of vaccines. Now, to maintain and build trust in science, it's important to engage with the general public and answer questions about what we do and how the scientific process works. It's the public and their representatives that will put policies in place that will move agriculture forward. Now, to this end, at UC Davis, we established the Institute for Food and Agricultural Literacy, and the mission is to create a community of researchers dedicating to making scientific information accessible and relevant and interesting. And we aim to provide the next generation of researchers with the tools they need to communicate science and infuse scientifically sound information into the public discourse. So I believe instead of worrying about the genes in our food, we must focus on how we can help children grow up healthy. We must ask if farmers and rural communities could thrive, and be sure that everyone can afford the food. And we must reduce environmental degradation. So with a shared vision and global cooperation, we can harness the power of genetics to alleviate human suffering and safeguard the environment. Now, I want to thank so many people that were involved in this project, and many organizations and funding agencies that participated. I am truly grateful for the support of my laboratory team and colleagues for this work, and only some who I'm able to show here. So I'm happy to, if there's time, I'm happy to answer questions. Thank you.
Thank you very much, Professor Ronald, for very, very scintillating presentation. And as you said at the end, it is exciting time to work on biology and agriculture, but I think it is more exciting to listen to you and to know all the developments in rice which have made significant contributions and changed the way rice research and rice science has gone into. I would request both of you, Professor Ronald and also Professor Datta, if you can take a few questions from our very learned audience. Kutub,
Yes, sir.
can we take few questions from the chat box, and then of course we can request if somebody have got another one also. So you can take up few questions from the chat box.
Okay, sir. Thank you. Yeah, so listening to these 2 evening lectures was wonderful for us, to the organizers and for the audiences, obviously. And I especially thank Professor Pamela Ronald for waking up so early for us, because it's around 6:20 a.m. there. So thank you very much. And yeah, there are a lot of questions. So I would go for the questions that is intended to ask to Professor Swapan Kumar Datta. Sir, BK Dash has asked that, Has anybody worked on mutation breeding for reduced phytate content in rice grain? And how fast or easy to screen mutagenized population for reduced phytate content? Although it's not very related to your topic, but you talked about phytate.
No, Kutup. I think it's very related to the question because I mentioned about low phytate and I mentioned also the mutation breeding. And definitely there are reports, low phytate barley is well established, low phytate barley is available. Low phytate rice also available from China. There are some sources, low phytate rice. But the problem with those kind of material, they're completely in a very different background. So when these background material are being utilized in mainstream breeding, then it's a really tough job. And so that's the reason even the Chinese did not go further, rather than they work for straight away taking the best variety and trying to look for the low phytate rice by genetic engineering. And mutation breeding, it takes hell of the time to screen for mutant line. And that's really one need to be lucky. When someone is lucky, one could get a line very quickly, but it needs a lot of screening, and then eventually you need to be lucky to find something which is useful.
Okay, sir. So the next question I'm going to ask is a very common question from a lot of publics. So this question could be answered by both of the speakers of this evening. So one participant asks that, are GMOs safe to consume, and any health implications upon long-term consumption? Also, this has been already answered by Professor Pamela Donald in last few slides, and also you talked about that. But still, it's good to discuss about this.
Okay.
Go ahead, Professor Datta.
Well, look, I have consumed golden rice myself, and I'm still doing fine. And last 20, 30 years, all commercially available GM crops, either edible or indirectly edible, even cotton oil, seed oil, also consumed. They're all scientifically safe, as well as from the consumer point of view, no adverse effects so far been found or reported. So it is as good as conventional breeding material. So it's not a GM particularly is safe or unsafe. They're the same as conventional breeding material.
And maybe I can just add to that. I think it's hard, especially for the general public. They don't know what scientists are doing. We have, for example, a lot of vaccine hesitancy in the United States. But we know for many, many, many years, right, that and every independent organization all over the world, that vaccine saves lives. And especially during this pandemic, it's really critical to vaccinate. And in the United States, there's some numbers that 40% of the public is afraid to vaccinate. And so we do really need to build trust in science. And this idea of scientific consensus and international consensus is critical. Most people I don't know, there are people that will accept the consensus that the climate is changing, but reject the consensus on plant genetics that the crops are safe to eat, but that you can't really pick and choose your science, right? You need to go forward and really have faith in scientific institutions that if there is a consensus that the vaccines are safe, they're going to save lives. If there's a consensus that BTA plant, for example, in Bangladesh is going to help farmers and enhance productivity and reduce chemical insecticides, it's really important that the public embrace science. And I think this is going to continue to be very, very challenging for all of us to really help the public understand that science is not just the opinion of one of us, that it's really a global community of independent scientists that have been studying these processes for many years.
Okay, so I think next, Professor Eki Singh from ICAR, he's the director of ICAR. He has a question. So, sir, you can straightly go ahead to ask your question.
Thank you, Kutu, for giving me this opportunity. Excellent presentation by both Professor Datta and Professor Pamela. I'm glad to see Pamela referred two slides from our work on golden rice and other on pyramiding of PI2, PI54. The first one is general observation that why golden rice in India has not reached the people. I would not blame the policy. I would not blame the regulation for this. I would blame the science. We spent 1.5 decades and 3 times we came out with the product in most commercial background in Sorna. Starting with the first event, then second event, SGR1, then with SGR2, and SGR2 we ended up finding the disruption of OX1 gene. And then the backup event, E event came, that only went to Bangladesh, it never came to India. So I think on golden rice front, if we have to blame regulation, we are wrong on that. Regulation is not to be blamed, the science is to be blamed for that. We have to have this, the government is changing minds, but we must have a product. Today, if it is open that bring the product for deregulation, I'm sorry to say that we will not have many commercially viable products which are available. That's the general observation. The second question to Professor Pamela, it's a very intriguing question for me. Whenever I see this the slide of sub-1 plot from IRI, where we have IR64 sub-1 versus IR64 normal, along with several other varieties. And you always find that you find some plants in IR64 are original variety. They pop up as healthy as the sub-1 carriers. What happens in these plants? Why these plants survive is something that I have not been able to understand. What makes them survive? They don't have sub-1, but they do survive. Has there been any analysis of these plants, especially at least 5-6 plants in each of the original variety? They do survive in the completely submerged conditions, similar situation. The third question I have, I just wanted to know how much area has gone into the rice carrying transgenic XA21, carrying transgenic sub-1, carrying transgenic PS stall. These genes have been cloned, characterized, transgenic plants have been developed. Why it is not happening that the transgenic, whenever we have a product that is the same gene from regular breeding program, it is spreading like sub-1, we have got 5-6 million hectare area, XA21 deployed widely, but the transgenic lines of the same genes, they're not really picking up in terms of area. If they are there, I'm not sure, you can educate me on that. So these are the 3 observations that I had to make. I will be glad to listen to your response.
Thank you. So yeah, some of you may have noticed in that really beautiful video that was taken at the International Rice Research Institute that the sub-1 varieties do fantastic. They survive the flood, they produce 3-fold more yield, but some of the varieties without sub-1 actually are also surviving. And for many years, it was thought it must be some quantitative trait locus. In the end, it turned out to be a single gene that was conferring this robust tolerance. And I think what the breeders, Dave McKill and others would say is that the fields are never perfectly flat. And so the water, the depth of the water varies over the field. And that you also, it depends on the weather too. So the plants are very, very sensitive. So you can imagine if in some part of the field, the water's maybe slightly lower, even without sub-1, the plants can survive. There are researchers looking for additional genes that enhance submergence tolerance, but so far those haven't been identified. So then your other question about the transgenic work. So we did that work in my lab. We isolated the gene. We engineered rice plants in my lab using a really a sort of a model variety called Lao Gang, and we showed that a single gene can confer resistance. But at the same time, we were collaborating with Dave McKill, who was using the sequences to develop markers. So the varieties that are in the field now in India and Bangladesh were developed using marker-assisted breeding. So there's no need for the transgenic XA21. When you're working with a rice gene in a rice plant, marker-assisted breeding is very, very powerful. Not only can you obtain plants that survive the flood, but there's no regulation, right? Because if it's marker-assisted breeding, that technology is not regulated, even though it's the same gene you bring in through transgenic. If it was genetically engineered, the regulation probably would have halted it. So there's not been any efforts to introduce the transgenic variety. It's not necessary in this case. And then I'll have to ask you a question. So the variety of golden rice that's been field tested, well, it's been field tested in Bangladesh. It looks like hopefully it'll move forward. Won't that variety be suitable in India, or is the genetic background not optimal in India? So you said you still don't have a golden rice variety that is going to be accepted by farmers. Is that accurate?
Well, that's I think in the BR29 background, and now there is a tripartite agreement between India, Bangladesh, Nepal, and this has been extended through IRI. So any variety that is released there can get automatically released after due testing in India. So that can be tested as and when it is cleared in Bangladesh, and also when we have the situation accepts it in the country, that can be done. But the same, it comes from e-event that can be used as a donor also to transfer into Indian background. That's not a problem, that can be done.
But Dr.
Okay,
Singh,
good.
sorry, please go ahead, madam, please go ahead.
No, you go.
Okay, no, but Dr. Singh, whether that treaty is applicable for GM rice also? That India, Bangladesh, Nepal treaty?
I mean, it can be released because release and notification is by same system of Seed Act, but it has to go through the transgenic testing procedure. Without that, it will not happen. It has to follow the approval from GAC, then only it can be done.
Kutub, there is one
yes, Professor Das, please go ahead. Please go ahead.
Ma'am, I was working at IRI with Dr. McKill, and our target was to integrate submergence tolerance with stagnant flooding. And that we achieved through conventional breeding. And as the genes controlling submergence and stagnant flooding, they behave in an opposing manner. That's why we achieved that one through conventional breeding. But my question here, although sub-1 QTL, which contributes about more than 70% of phenotypic variability for submergence and has been successfully introgressed into different mega varieties like IR64, Sierang sub-1, and BR11 sub-1, and Swarna sub-1, and Sambama Suri sub-1, and CR1009 sub-1, so on and so forth. Many mega varieties it has been successfully introgressed. But I could find at IRI that why, although the same sub-1 QTL has been introgressed into the different mega varieties, why in terms of survival and regeneration that varies? That in my rating, in submergence tanks at IRI, I could find IR64 was best, followed by Sierang sub-1 and some other varieties. Why it happens? Although the same sub-1 QTL introgressed, and it's a major QTL.
So Professor Das, I think it broke up a little bit, but I have a beautiful slide from Dave, and maybe you were involved in this project, showing sub-1 introgressed into many genetic backgrounds, and then after a flood, that the plants do very well with sub-1. But I think you had a question that I didn't hear, that there is some other effect of sub-1 in some genetic backgrounds. Could you tell me again what it was? What your question is?
And is it the role of sub-1 or the background history of the crop variety?
His question is that sub-1 does not behave same way in different genetic background. It has differential behavior in different genetic background. Some cases it works very well, other cases it doesn't. So is it a sub-1 into background interaction that is happening and that results into this kind of phenotype?
Oh, okay. So I don't know the answer to that. I think that's really your work, and David McKill, the beautiful work I'm familiar with shows sub-1 conferring tolerance in different genetic backgrounds. I did not know that there were some genetic backgrounds where sub-1 did not have a robust effect. So maybe we could talk about that after, because I'd love to hear more about that.
Oh, okay. Then when I asked this one to Erie's physiologists, they told me that some minor QTLs contribute that. But I was not satisfied. I told, is it the minor QTL or the background history of the crop variety? Say, for example, ICAR-64, the slide Dr. Datta showed, that so many parents were involved and in the development of ICAR-64. So in the genealogy, that might have helped rather than the sub-1 QTL alone. I don't know, that's why I'm asking.
Mm-hmm. Yeah, that may be. There may be other QTLs that are critically important that we just have not yet identified.
Thank you.
Thank you.
Okay.
Thank you very much. Kutub, can we take maybe one or two more?
Yeah, we have a lot of questions now. So yeah, we can take. So next question, there's two questions from Rupam Kumar Bhuya. So he wants to know what is the yield potential of golden rice? As the question goes to Professor Shapan Datta. And what is your take on rice bran oil? So this is the question.
I did not hear the second question. The first question.
Oh, second question is not also related to your talk, but he wants to know what is your take on rice bran oil?
Bran oil, okay.
Yeah, because you talked about nutrition and all those things. Yeah.
Okay, the yield potential, I think the golden rice yield potential remains same because BR29 when we genetically engineered for golden rice, as well as we developed also several other indica background of different Asian countries. We didn't see any changes, but the AK Singh's experience is different. He found several strange situations, but that's a different thing because he got material from IRI, but we developed our own genetically engineered material and we used anthraculture derived homozygous line. So our material developed by in a different way, and we did not find any yield penalty of golden rice. So from yield penalty point of view, that is the answer. The second question is bran oil. Well, bran oil, although I'm not sure what the
it is. I think it's a general question on bran oil. Is that Kutub? So if
Yes,
if I can take that,
yeah,
the total bran oil, potential in India is 2.4 million tons. If we process the bran from rice, currently we are producing only 0.9 million tons. So there is a huge potential to harness the bran oil from rice. There are a number of companies in India which are marketing bran oil from rice and especially from basmati rice. There is a brand called Basmola. Basmola is the oil from basmati, bran oil from basmati rice is Basmola. And there is difference in the fatty acid profile of basmati bran oil versus non-basmati bran oil. We are doing detailed analysis of this composition also. And the only problem with bran oil is stability. Once you do the milling bran oil, because of lipoxygenase enzyme activity, it becomes rancid very fast. Yeah. And we have isolated some mutant which are lipoxygenase null. Now we are trying to transfer those genes through marker-assisted breeding in the commercially grown variety, so that there the lipoxygenase activity is not there, and bran gets stability. Even after milling, bran can be stored. Otherwise, currently, soon after milling, you have to extract oil from the bran within, say, a matter of 20 hours. Otherwise, it will become rancid. I will not be able to process it. So there is a huge potential, excellent quality oil in terms of fatty acid profile. And this has huge potential because we are importing total oil of 77,000 crores annually. If we add 1.5 million tons of bran oil, high quality bran oil, that will also substitute the import bill. Thank you.
Yes, sir. So the next, I think this is an interesting question. So from Shashank Maurya, and he asked why we have not thought of an exhaustive effort on social science research on regulatory aspects of GM crops.
There's a lot of social science research being carried out, and it's absolutely critical. I think that in a sense, the scientists have done all we can producing really useful tools for breeders, and much of the adoption will depend on how the public sees the technology, and social scientists are very important. I'm part of a committee at the National Academy of Science, which is primarily social scientists that are really studying ways to communicate. They're studying the science of communication. And one huge aspect that social scientists are looking at is the spread of disinformation and misinformation. So disinformation is information that is known to be incorrect, but is spread out for, really nefarious purposes, often to make money. And so this is a huge, huge problem. Social scientists in this day and age have a lot of work to do. Obviously, a lot of them now have shifted their efforts from plant science to vaccines and trying to understand this vaccine hesitancy. So this is going to be a challenge for all of our lives, I believe.
Okay, thank you.
Professor, can I add one sentence on this?
Yes,
What
yes.
Professor Moria asked. As a matter of fact, there is a lot of anti-GM social science research, and Professor Moria wanted to know whether there is pro-GM social science research happening, and unfortunately not much. And that's one area which is very, very important in country like ours, but anti-GM social science research is very strong. It is doing great job as far as their agenda is concerned, but we are lacking on that communication, and we have to do a lot, I think.
Okay, thank you, sir, for your insight. And there are a few more questions, sir, if the chairs allow me, so I can continue with some.
I think very quickly, you can take one or two. It is 52 past 8, so maybe another 5-6 minutes we can continue.
Okay, sure. So there is one question that has already been answered, but I'm not going to ask that question. So, madam, what is the validated gene or QTL for stagnant flooding, which can be integrated into popular varieties to develop both stagnant and flush flooding? Yeah.
So that's the work of Professor Das, actually, and maybe he can take this question. So the question is the work that you did with Dave McKill to integrate another gene for stagnant flooding. I don't know if he's still there, but he did mention that work has been done. I don't know if those varieties are available to farmers yet.
Yeah, okay. So on a similar note, there is also a question from Abhishek. He asks that, is there any QTL that confers double resistance to saltwater inundation? It may submergence plus salinity tolerance. So because in this region, farmers are mainly facing this kind of problem because of the contamination from saltwater, because of the flooding.
Yeah, often when there's flooding, there's also salt, higher levels of salinity, and groups around the world have been combining, well, one gene's called salt tolerance, and that has been combined with sub1. I believe those have been field tested. Maybe are in the hands of farmers, but I'm not an expert in that work. Maybe some of the other participants have more information about that. But there is ongoing work about trying to identify other genes that confer salt tolerance.
Okay, so this is the last question, sir. Pathak sir, this is the last question I'm going to ask. So this question is from Niloufar Karim from Bangladesh. So she asks that I am interested to know if the high protein rice are aromatic in nature, and some Bangladeshi and Iranian varieties show high protein linked with aroma. This question goes to Dr. Sir. And
I think A.K. Singh also can comment on that probably, because he has been working on aromatic rice breeding. The material that NRRI reported that has no linkage, but A.K. Singh can tell because he has been working on some of the aromatic rice breeding, so that whether he has seen the protein levels and any linkage with protein and aroma.
To the best of my knowledge, no. The aroma gene is on chromosome 8, FGR, fragrance gene, that is responsible for 2-style-1 pyrroline. There is another QTL for aroma on chromosome 4, but they are independent of protein content QTLs, which have been mapped in this line from Assam rice collection. I think Dr. Chattopadhyay, if he's there, he can tell where is those QTLs located, but I think they are independent. There is no linkage between that. And
Sir, actually, so I'm Krishnendu Chattopadhyay speaking.
Yeah,
so actually, this chromosome 1, that QTL is located. This grain protein content 1.1, this QTL is located on chromosome 1, and we have not find any kind of linkage with aroma. But report shows that there is some linkage with iron zinc content with aroma. And actually, this has been reported earlier by Dr. Sharma in our institute, that there is some kind of association, phenotypic association actually, between this iron zinc content and also with the this aroma. So, but still there this also reported that this iron zinc content QTLs are located in different chromosomes, not in the same chromosome where the aroma is present. Thank you, sir.
I can also add to this point that to confirm that iron and zinc, they have some association. And at the same time, even transgenic rice with ferritin gene, we have seen that enhanced zinc also in the transgenic material. So there is some linkage with iron and zinc, and that's true.
Okay, so thank
but
you.
One thing I can say for sure that now with genome editing, any variety can be made aromatic because they all carry, you know, it's a recessive gene, and all varieties, non-aromatic ones, they carry dominant genes. So you can have a very targeted creation of aromatic variety in any variety which is high yielding can be made aromatic with genome editing in one go.
If it is required.
Okay, good. Sir, the last one, maybe in yes or no. A question from Dr. Shekhar Nayak, whether golden rice has got shorter keeping quality.
What quality?
Storage, shorter. It can be stored for a shorter duration. Not
golden rice. What I mentioned in my talk, that's the reason we used reducing the lipoxygenase gene, that enzyme that makes the rancid. So for a shorter period, definitely golden rice is okay, even in room temperature. But after harvest, if you keep it longer period of time, like any other rice, if you keep, then the carotenoids degradation will take very fast, and every month carotenoids would be reduced to a greater extent. So I think this is one bottleneck that is the high carotenoid rich rice. So after harvest, one has to use it as quick as possible. Unless you make it more effort to make it double haploid as well as lowering the lipoxygenase.
Okay, sir, thank you very much. Of course, every good thing sometime comes to an end, and this is same for today also. Now I request Dr. Maiti to propose vote of thanks. Dr. Maiti, over to you, please.
Thank you, sir. So, respected eminent speakers, Professor Shravan Kumar Datta and Professor Pamela Ronald, Chairman of the session, Dr. Himanshu Pathak, august audience, ladies and gentlemen. You will all agree with me that it was a real evening treat to all the rice research workers through the deliberations made by Professor Datta and Professor Ronald, which he just listened to and shall be imbibing the knowledge and thoughts radiated by both of them so lavishly, which will pave the way for our future research roadmaps for nutritional and overall food security through rice in this planet. So on behalf of the whole rice family and my own behalf, I take this opportunity to express our gratitude and thanks to both the eminent speakers for sparing some of their very valuable time for such thought-provoking deliberations, and that too in some odd time for both of you. Thank you very much. I extend our sincere thanks to our chairman, Dr. H. Pathak, for kindly chairing the session very successfully. Finally, on behalf of the Association of Rice Research Workers and my own behalf, I extend our thanks to all the dignitaries, distinguished delegates, press and media personnel, all ARRW office bearers, and the technical support teams to the virtual platform for their enormous contribution to make this evening session of the first Indian Rice Congress successful. So it was a very exciting day today, but the only regret is that it has come to an end today. So good night.
Good night. Thank you very much, all of you. So see you tomorrow at 10 o'clock with another sessions and good discussions. Thank you very much, all of you.
Thank you very much, Professor Datta and Professor Pamela Ronald, for excellent lectures.
Thank you very much. Nice to see you all.
Nice to see you. Thank you very much.
Thank you very much. Thank you very much.
Thank you, sir. Thank you.
Thank you.
Thank you.
Thank you, sir.
Thank you, Kutub. Thank you very much.
Thank you.
Thank you, sir.